Why is methane interesting? Maybe you may be asking me now. Well, methane is nice food for a number of microorganisms. Hi, Lisa. Thanks for being here, by the way. Methane is used by a number of microorganisms that live in these deep sea areas, and they can process it and transform it to organic. So as plants use photosynthesis, thanks to the sun in the outside, right? These guys are able to transform methane to organics, and then they are primary producers for all type of animals that we're looking also for. So the hope that we have is that you are seeing a lot of animals right now. If we get to see those methane seeps, then you have the primary producer for a whole ecosystem of the kind of species we are looking for. So to speak, if we find such methane seeps, we maybe find a true garden of Eden of species that we hope most of them are yet to be described by science. So at any time, please, if you have any questions, just tell me in English o también español. No hay problema que sea en español también. Aprovecho de aquí saludar a mi hija Margarita, que me está acompañando aquí junto con su conejo. Me está mirando aquí en el teléfono, viendo esta actividad, y recordarles a todos que la ciencia es para todos y todas. Tenemos la suerte de tener muchas investigadoras en el equipo, siendo la ciencia y la investigación, como digo, algo para el alcance de todas y todas. I will continue now in English. Now we're going as fast as we can back to an area we just detected that had a lot of rocks. And we are looking also for rocks because a number of those living fossils, as I said, these very ancient species need and like a hard substrate. So when we are looking for the methane seep, we just detected a patch of rock that seems to be colonized by a number of different species that we're coming back as fast as we can, as I said, in order to try to take as much samples as we can in order to say whether any of those little shells of every kind may be among the species that we're looking for. Among others, for example, brachiopods. Brachiopods are animals with two shells similar to mollusks and bivalves, but not the same thing. They're similar, but the interesting thing about brachiopods is that most of the species that we know are in the fossil record. You may find even, for example, fossils of brachiopods in eBay or Temu or places that you can find such general things. But in this case, we are looking for the living relative of those fossils in this area. That's why we wanted to come back to this rocky patch, because when we went through that, we saw some of those, which also include chitons. Chitons are also very ancient animals. In this case, they have like a armored plate on top of them that protects them from potential predators. And then we are looking for such relatives of those chitons that we also know from the fossil record. Also with me here are Guillermo and Nick. They are the true experts on all the deep sea biology that we have around. So at any moment, Nick or Guillermo, if you see something interesting around to comment, just you can speak and give me details. So Guillermo is telling me right now is that we have seen eel-like fish. We just passed it over. Also, we have seen at least 5 different type of species of stars. And then between yesterday and tomorrow, more than 7 types of sea cucumbers. We have collected some of them. They are from every color and size. Some of them green, some of them red, some of them deep purple. And I have a very question now from Peter. Thank you, Peter, for your very good question. What is the primary source of food for organisms outside the methane seeps? And then, do you expect to see any obvious morphological changes in organisms that exist in the methane seep ecosystem? Actually, that may be actually the case, because, as I said, some of these guys, they are living from methane, and how they do get the energy is that they either eat the microorganisms that are using methane as a source of energy, and more interesting yet, they form an association with such microorganisms. So they along the process you may know as symbiosis, right, in which two different kinds of species, in this case a microorganism and an animal, an animal provides the place where they can live, protect their environment, to feed from the environment, and then the microorganisms in return they give you food in terms of the methane they produce and then transform to organics. And about obvious morphological changes, maybe Guillermo or Nick may comment on whether we can find such adaptations to this kind of environments. Also, Guillermo is commenting that there's many animals with that are attached to the bottom, like corals, sponges, sea lilies that are crinoids. Crinoids are interesting again because we know many species that are in the fossil record, and we have already seen and collected a number of crinoids as well here. Again, with the hope that we are collecting new species and hopefully also very ancient species. And these species typically live in a way that they are just attached to the sea bottom, waiting for whatever the sea currents are bringing to them in terms of organic. So they are just sitting there and waiting for whatever just by chance they get, and that's the way they live. So that's a kind of adaptation that's maybe related to Peter's question. Also, Guillermo comments that environments like these methane seeps, they contain animals like giant clams that, again, they are able to survive because in this case they are in symbiosis with the kind of what we call, actually the name is methanotrophic bacteria, that is, they are able to eat methane, so to speak. So along other species. And then one way to detect ancient seeps in the area, if we find them, that would be on places where you can find the remnants of those giant clams. So if we see a place where you can see only the shells of those clams, not the animal itself, that could be a sign of an ancient methane seep that is not active anymore, but at some point it was. Then I have a question on near north. Why should this methane seep associated to the rocky patches? Actually, that not necessarily is the case. It was just a chance finding. When looking for the methane seeps, we went over the rocky patches, and all the deep sea marine biologists jumped in their seats, seeing all the colonization that such rocks had. That's where we're coming back to that area. I don't think there's any association between rocky patches and methane seeps. It's just because we went over it just by chance.
I want to make a comment. The primary source of food for organisms outside the methane seeps, detritus. Okay? Most of these organisms are detritivores, most of the echinoderms. But also, of course, we have some predators. Okay, so there's plenty of those types of organisms, okay? That exist in methane seeps.
Also, Guillermo is commenting that in this methane seep, typically you will find tube worms that in some cases, these tube worms can be really big. In other areas, they can be up to 20 centimeters, even half a meter long. They can be huge. There's a number of tube worms that we can find around. So again, if we send the remnants of such tube worms, we could be in presence of an ancient methane seep. And maybe Nick, you may comment on the things we are looking at it. It's, for example, I see some corals, something like looking at anemone and so on.
The pink reddish organism was a crab, if I'm not mistaken. And this is one of the corals, the white corals. But the true diversity, here we have a crinoid coming up on slightly to the right. But the true diversity of this environment is within the sediments, especially the upper 5 centimeters, especially if there's oxygen. That's the, we call this myofauna. These are small organisms, many different phyla, and but unfortunately you cannot see them with your bare eye. You need to work, you have to see them under the microscope. But it's a fascinating group of organisms.
Guillermo, you may also comment whenever you want in Spanish. Maybe you can sit along Nick.
We're coming up on a white sea cucumber.
Bueno, muchas de estas especies son oportunistas, ¿ya? Entonces podemos ver estos mismos camarones en distintos ambientes. Lo mismo en el caso de los distintos peces que hemos observado. Estos viajan grandes distancias para encontrar algo de alimento y muchas veces están detenidos en el sustrato porque el costo energético que tiene desplazarse es muy grande y la cantidad de alimentos disponible no es tanto. Entonces, cuando ocurre algún caso, por ejemplo, de lugares donde hayan ambientes de emanaciones de metano, esto se vuelve tan rico, es un verdadero oasis de organismos que están ahí, digamos, dependiendo de estas bacterias. Eso hace que obviamente estos puntos bullan de vida y con distintas especies. Lo mismo ocurriría en caso de que apareciera algún animal muerto. Eso va a atraer animales de todos los lados y generar por unos breves meses todo un ecosistema asociado a la descomposición de algún animal.
I have a question there for my daughter. Have you captured any chimp yet? Believe me, we have tried several times, but they're so damn fast that we have been able to. We'll keep trying. Actually, I see we got one just by chance because I have to confirm that I think that when we were taking a rock sample, one of them were clinging on that rock. So maybe they came just by chance we got on one of them. But I will let you know. And now we are taking a core sample. That is, we are going to insert one of these tubes into the sediments in order to. We can do a number of things very interesting with the sediments here. So the way we do that is that the robotic arm from the ROV will take one of those containers, which is called a push core. That is, you can push it onto the ground itself. And if we are lucky, the core, which is the sediment where you can see in the background, they will not fall off. They will remain in there. And from that, you can do many things. For example, you can characterize all the geology on the site. You can do many things. For example, you can characterize the geology and then characterize all the geology and mineralogy of that. You can also inspect what all the tiny animal that lives in those sediments may be. You can also characterize all the kind of biosignatures. With that I meant all the type of biomolecules that we know that are related to life. So if you're looking for evidence of life elsewhere, for example, in the expected oceans we may find, we suspect they may be, in the icy moon of the solar system, like Enceladus or Europa. Why would you look for evidence of life? It's not evident, right? Like a thing swimming just in front of you. We look for such a biosignature. So we're now seeing that we are returning the core into this place here, but we know we need to use one or two because the researchers, which is a partner, researching for the Universidad Autónoma de Chile, it has to be between one or two because he's looking for light-sensitive microorganisms. So those kind of sediments must be among the first to be loaded off the ROV once it get up into the chip. So that's why we're changing now the push core. So now we're going to see how actually we do that, in which the robotic arm takes the core, pushes it into the ground as much as we can get, and then take back the sample into the container of that core, again to be inspected on the lab. And then here, let me tell you that actually the sampling process compared with the scientific process that follows, it will be like 1/10. I mean, we spend here 15 minutes maybe taking that core, but then you will probably take entire year in order to describe all that core can tell you about the site we're inspecting right now. Yeah, actually, Melissa is also commenting here. Yeah, they are James are kind of making fun of us because they tend to swim just in front of the camera, telling you, "Blah, blah, we're here, you cannot catch us." So let's see here how we can take that sample for our partner.
I wanted to mention that the tremendous effort it requires to describe a new species. It may take months, 2 to 4 to 5 months to describe a new species, just based on a few specimens. So 20 minutes of sampling, 5 months of work. I'm not too far off, okay, from the reality here. So just to get an idea.
And then you have to write all your results, and then you have to publish, in which the manuscript, as we call it, it have to be reviewed by your peers, the expert in your field, in order to say that everything is correct. And so that takes another half a year also. So it's a lengthy process, science. But what you gain is tremendous. They describe this beautiful life that we have on Earth. Margarita says also, have you guys found a new species? Actually, we think we have already found many. And that's why we want to come back to the rock analysis as fast as we can in order to see what we can find there.
Yeah, this is I think there's a rocky...
Yeah, Aparna, if you are hearing, it's kind of rocky there, so you won't get much. You wanted to try...
We'll just keep it in our arm and keep flying and see if we can find a
Okay,
opportune place
yeah.
to grab it.
All right, let's keep moving and then see with something that it looks less rocky below that sediment.
From where? She's 506. You're about... Yes. Waypoint 7 is... Which one?
How do we access this chat?
You gotta give me a coordinate.
Okay.
It's supposed to be just already given the coordinates?
Is it waypoint 6?
Yes. If you find them on the coordinates of the rocky types? Yes. They gave it to you, so they gave it to you. Yeah, let's go back in the same group.
Go backwards?
Yeah, let's come back to the same group because we want to come back to our rocky patch we just passed when still Miguel was here, so as fast as we can.
So sorry, I'm not following. So there's a rocky patch, but I just need a coordinate where we're going because I'm not sure where we're at here. So is it like we've got waypoint 6, waypoint 6.4?
So continue on the first comments of Guillermo. If you could have at least one potential species of a new crustacean. It was collected a year associated with a sea spider. Those sea spiders are amazing to be seen, by the way. They actually look like eerie spiders of the deep sea. Maybe Nico also and Guillermo may comment on the other amazing things we observed yesterday or today.
Say that again, Armando, to make...
Maybe you can comment on the amazing living thing we saw yesterday or today. The one sample that you already have or things you have seen during this dive or yesterday's.
Well, we have been able to collect many specimens, but that was just the first day of the expedition. So we do find typical groups of animals that we expect to find at that depth, but obviously we're not certain of the exact species or the exact genus that will be determined during the lab, during detailed examination in the lab.
Copy that. Come to the top. So I'm going to. I don't think I'll just try. Oh, it's here. It's already there. Yeah, you see it? Okay.
So now the ROV pilots are calculating the coordinates, so the rocky patch we want to come back to in order to inspect them.
Let me get moving there.
And again, why we're coming back there? Because as we went over it, we saw the high colonization of different type of organisms like corals and brachiopods maybe. We also noticed a few chitons, so we're hoping to come back to that exact site in order to sample as fast as we can with the remaining time we have.
Yeah, no worries.
And then Matthias Portcliffe saying hi, so hi Matthias. And I love the three little cats he just put before him.
You want the core there? Okay. So maybe what we do is that while he's looking, we can find a place.
For the core. Oh, the core, yeah. Maybe here. And since we're going to get to a rocky patch now, right?
Try it. I don't think this is a great spot, to be honest with you. It's, I've just put it in there and it's...
Yeah, whatever we can get.
Yeah, maybe we'll down on the flats or when he moves,
Okay.
we'll find a good spot down there.
Okay. Maybe Guillermo or Nick can comment what is that white thing that is just flowing by?
What are we?
No. It is? Yeah, it's much nicer.
So Guillermo is commenting here that why we're coming back to that patchy rock, because in this kind of soft sediments, there with a lot of organic material, is sessile animals, the ones that like to live in one place. They cannot attach themselves. That's why they're looking for rocks, which rock themselves are also contain a number of little critters, so to speak, including the type that we're looking for, like brachiopods and chitons. That's why we're coming to look for rocks as a preliminary approach to get to the animals that they live on and in them. And also Margarita is saying, yes, that's exactly. She say, well, it's like when we look for chimps in the seas of the Animal Crossing game. That is the case. But this is the real world. We're looking for actual animals. That's correct. Oh yeah. Austin says it's hard to pay total attention for 9 hours of a dive. Yeah, you're right. But let me tell you, when you are here and in front of the screen, knowing that actually that is happening just below you, it is difficult to lose your attention because everything from many of us here is new. Yeah, Lisa also comment how you will collect the things that burrow so deep. Well, most of the time, just by chance, that when you get those one of those cores, just by chance you collect the number of living things that were in those sediments. And then, Nico, you can comment us on the sea cucumber you see at the left.
I was going to comment on first on a comment that I see on the chat. It says there's a comment saying that I would assume that the organisms are primarily concentrated near the vent seeps. Well, it is correct if you're talking about macrofauna. If you find an active seep, you will find a lot of, for example, bigger crabs, mussels, clams, the tube polychaetes of the family Cyboglinidae. But as you move away further from the vents, there's still life. Even though you see this type of sediment right here with an occasional shrimp, it's actually teeming of life. It's just the size of the animals is much smaller. So there is life away from the ships. And yes, you see low density of larger organisms. This is very true. But again, there is what we cannot see is the millions, literally the thousands and millions of organisms that live in the interstitial space. How do they survive? Well, the place is full of detritus. You have all types of feeding modes. You have detritivores. You have predators. You have commensals. So there is a lot of life. Some that you see, some that you don't see. We're coming up on a sea urchin to the left, kind of a dark color. Let me verify. Yes, that is a sea urchin. Yes.
Also, Guillermo's commenting here that since these sediments are really poor in oxygen, what is happening that many animals make these little tubes, tunnels, so to speak, in the sediments. With that, you get water that has some oxygen into those sediments, and then when you have that, then you will have a biofilm that is a thin layer of microorganisms that grow onto these little tunnels, in which you can find, in this case, bacteria that are like to be in an environment with oxygen, but in a sediment low in oxygen. So if you understand the idea, again, you will have the sediment that are low in oxygen, but you have animals making little tunnels that carry water with a higher concentration of oxygen into those sediments. And when you have that, you have a colonization of microorganisms that are able to live in an environment with more oxygen.
So this is a foraminifera. These are the, how do we call them? The foraminifera? How do we call the group? The group, what's the name? It's a biology something? Oh, rocks.
Also, we can see some brittle star. Maybe Nick or Guillermo can comment us on brittle stars, the little white, the one actually there is one walking just down us, below us.
That's right. These are all detritivores, echinoderms. One of the 5 classes of echinoderms, very common in sediments and including the deep sediments. I just want to mention that the sediment is highly. It's not undisturbed. There is a lot of biological activity evident by the mounds that people see. You see one of the legs is missing there. They can autotomize. Right? The arms. They can autotomize for as a defense mechanism. Their arms, and they can regenerate them.
And the other thing you can see here, it is a comment by Guillermo again, is that you can not only guess on the animal that live in a particular site, but also on the trails they live. So he just noticed a number of trails that were made by sea cucumbers. And then also maybe Nick can comment on what we are looking at now.
The trails are made from sea cucumbers, mainly sea urchins, ophiurids. Here we see that looks like an acorn worm in the middle of those two. No, I'm not so sure if that's an Ascidian. Well, but we see here an outcrop of rocks that we may want to examine, Armando.
Here, right now? Or you want to wait for the rock garden?
No. Let's go to the rock garden. I did not know there is a rock garden.
Okay, yeah,
No.
we're going to as fast as we can. We are going as fast as we can to the rock garden.
No tenemos solamente escudo. El arma mecánica, lento.
So again, this area in particular here may look like this is a wasteland, but it's not the case, because again, there is so many things you cannot see because they are living just below the ground, and also because you cannot see them because they are too small. For example, microorganisms. Maybe now we are seeing like a little squat lobster. That's the name, right, Nick?
That's a squat lobster to the left. Yes. That is correct. Yes. And a kind of a whitish or translucent sea cucumber hiding underneath the rocks. You just have to pay attention. It's actually full of animals. Let's see what we are coming up to. Could be possibly a city. Yeah. But the more you look, the more you will find.
About the last question about if there are differences between the night and day, no, there are no differences. You have to think there's no light at this depth. So if we just shut off the lights of the rope, this will be completely dark. And also the temperature is quite stable. There are not big differences. And it is about 2 degrees we were measuring this morning, so it's quite 2 degrees. So it's quite cool. Well, so yes, you have to imagine this complete darkness. Yes, very, very cold.
And there's actually a question here that this is of my ignorance. Where you have light, most life on Earth have adapted to have what we call a circadian rhythm, in terms of when you know and how you accommodate your biology to day and night. That base where you can with your different sensors, in the case of your eyes and the temperature in your skin, etc., you can tell what is day and night. So question for Nick and Guillermo. In this
we see here
do animals have a circadian rhythm or not?
If the animals have what? I'm sorry.
Circadian rhythms here, so deep where you have no sunlight, or they have a resting period during night, so to speak?
They have chemical cues. By far, chemical cues here are what the organisms are using to find each other, and also sounds. Sounds, bioluminescence. Bioluminescence is well known to occur in deeper areas. Anywhere from predator avoidance, recognition of species for mating purposes, So there are other types of communication. You don't need light to communicate.
And-
Life has found ways.
And Nick, the question is, do animals sleep here?
Well, if you want to collect this, Guillermo, we were looking at the purple starfish to the left, and Guillermo would like to collect it, if it is possible.
Oh.
And now we're going to try to collect that star we just
Guillermo
saw.
is a professor of.
Understood. Coming to a stop.
While the sediment settles here, as you may see, every time the rover goes into the sediment, it tells you about how loose that sediment is. That's why you find this cloud of sediment flying all around us. Let's answer Marita's question, where she wondered if we are looking, what is the influence of pressure in this depth? What happens to the body of this animal when they are brought to the surface, Nick or Guillermo? Again, a question on Nick to Guillermo, or maybe a Victoria. What happened with this animal when you bring them from this deep into the surface?
Okay, they are asking what happens with these animals when you bring them to the surface. ¿Qué pasa con estos animales cuando se les trae a la superficie?
Bien, muchos de estos animales, claro, crecen y se reproducen y hacen toda su vida a estas profundidades. Por lo tanto, están muy bien aclimatados a esta presión que tiene el mar y también a las temperaturas tan bajas que podemos observar acá. Una vez que están arriba, muchos de ellos colapsan, algunos debido a la presión interna de su cuerpo. Pero en algunos casos, como las estrellas y los erizos, debido a que tienen un endoesqueleto que le permite una mayor sujeción, pueden soportar estas condiciones. En algunos casos sí, en algunos casos no.
¿Y qué sería lo contrario, Guillermo, cuando llevas un animal a la superficie a estas presiones?
Ocurre una implosión de los organismos. Si nosotros bajáramos desnudos al fondo marino, nuestro cuerpo no toleraría esa presión y obviamente se vería aplastado por todos lados y como una verdadera implosión.
So what we're now trying to catch is that star. We're trying to get that starfish now. The way we're going to do it is to use this like vacuum-like instrument in order to suck it a little bit, just enough to grab it, and then put it in what we call a bio box. That is a box designed to collect organisms like this one. The idea is that this starfish seems to be so big that in order to get it, we need to use such a strategy. If it were a smaller starfish like the brittle star we just saw, we would use the suction mechanism that you see here. This is just like a vacuum, which directly connects you to a jar that little star would go. But in this case, it's so big that we're going to collect it in this way in order to suck it just a little bit in order to put it in one of these collecting boxes.
It's going to be the box? Which box?
Como la
Sí, no, ¿cuál?
en la So let's see how this works. And again, here we have a number of persons that have been doing this for a long time, so they know how to do it. So now suction is being done gently, as you see. Then the whole animal can get into the box that will get it until we can take it to the surface.
We still have the core on the other arm. There's the core, yes.
El core, la palma, está en la mano.
And we could attempt the core here because Aparna just needed 5 top centimeters. If we get that, that's good enough. She's looking for various. So we also try to get get a push core here. We attempted it before, but we didn't get enough. But were just told by the person that is looking for us that once that core that she just need the top 5 centimeters. So as much as we get that would be perfect. And yes, Lisa, you're right. We have great collection pilot. We usually have a lot of experience on this kind of sampling.
Well, as we are waiting for our wonderful team to take the core, I wanted to mention something about what we were talking before about sleeping and this animals' lifestyle. You have to consider there's not a lot of food in here and the temperatures are so low that they usually have this low metabolism. They don't usually move that much. Most of them are sessile or almost sessile, so it's not the kind of lifestyle we are used to, like super active in the day and relax at the night. So yes, it's a complete different life to the one we are used to. And well, with this I'm going to say goodbye and enjoy the rest of the dive. Thank you.
So again, now we're going to take another push core as far as we can with the aim of just getting the 5 top centimeter, because the researcher that is waiting for that core is intending to analyze the top surface bacteria that may be living in such sediments. So it doesn't matter how deep it goes as long as we have the 5 top centimeters, which is actually, it looks like it's already the case. Oh, I fell down.
It's pretty hard.
Oh, it's too hard, right?
I just want to make a comment for formal discussions of our findings. Please check our websites. There are several professors here, so you can see many published papers in highly regarded journals. If anybody wants to look at the end result of this type of work, where things are formally discussed there and they have been peer reviewed. Thank you.
Is that a rope that they left?
That looks anthropogenic.
Yeah.
Can take it. Very good.
It looks like the end loose of a cable or something.
Yes, but we gotta do some sediment course. That's gonna take too much time. Armando, remember, you still have a core on the left arm. Okay.
Yeah, let's try one more time for the core here, and we whatever luck we want, because also we want to get to the patchy rock. This will be our...
Nick, just to confirm, this is the push core number 2 that they didn't.
Yes.
So, okay.
Oh,
Push core
Yes.
number 2 for Aparna, if
for
it is
Aparna.
that possible.
yes.
detail we need.
Maybe Nick or may comment on what we are seeing swimming just by the core on the right side. Nick,
Yes.
what is swimming just above?
The floating, I can tell from the swimming, it's a sea cucumber.
That's more than 5 centimeters. You happy with that?
Yes. Oh, yeah, that's perfect. Thank you.
If we, there was a suggestion from Guillermo for us, if finish with a core, and I would like to take also a core on top of one of those mounds. Oh, yeah, yes. Let me see. After the thing settled. Yeah.
Then you wanted to take another core here as well?
Yes, we're looking at any mounds that result of biogenic activity.
Sure, like the little.
It's like a little hill.
Yep.
Just yes.
Say 12's okay for you this time?
That's okay. Yes, I don't have time sensitive material.
Okay.
Any mount will do. I will know the success after the fact. After you spend time in the microscope. One of the issues with the organisms that live in the sediment is the oxygen availability. And what we're trying to do is, at least for my samples, so we're trying to pick up the first 5 centimeters of the sediment because oxygen gets depleted very fast after just a few centimeters. So that's why you will see here, I'm looking at, we're trying to place the core in one of those mounds. Not necessarily this one, but oh no, that was the previous core. Huh?
Do you have a mound in mind that you're looking at?
Right now, I cannot do it with my eyes, to be honest. Like,
I'll give you a little zoom here. And this also will help you get a little idea of this. We can also fly around and kind of get a better, like if you see something along the lines here.
I don't want to take too much time. I mean, for example, this looks like a mound.
I suggest let's keep moving, and when you see one, we get down.
Okay.
Yeah.
Right. Those little mounds.
Here we have a series of
Okay,
mounds.
those little mounds. Any of those.
Yes. This is freshly for the people that are hearing us. These are freshly disturbed sediments. Okay. And of course,
We're going to kick up the viz because it just popped out of the dirt, so it's going to roll right through. Okay. We just could go 100 meters or something. We'll find a good one here for it.
un core para mí es un mount, pero no el mount que
So now we're looking for another push core, in this case for Nick. Nick Skizas is researching for the University of Puerto Rico. He's looking for these little mounds that are telling you there are fresh sediments that are being brought to the surface of some living animals. So that's why he wanted to go into the core with the, with the hope of catching those little animals that are making the little mounds. I have a question where, yeah,
Okay.
any of these, yes? I have a question here, whether we will find new anaerobic bacteria. Probably most likely. As I said, in this, Guillermo commented that this, or Nick, I don't remember, that these sediments are typically poor in oxygen, so you will find most likely anaerobic bacteria. What do you mean anaerobic? That means that are bacteria that live in an oxygen-poor environment. So they dislike, most of them, oxygen. They prefer to be living because of their metabolism in an environment where you don't have oxygen, which is interesting comment because that's how life started on Earth. We think that life started in an oxygenic environment, that is an environment with no oxygen, until the first life form, which are typically the type of bacteria that are able to do photosynthesis. We know they're cyanobacteria. They start producing so much oxygen in time that they completely change the chemistry of Earth. So then that life again has to adapt in order to live in an oxygen-rich environment. And so, but we still can find many environments on Earth that are poor in oxygen, where all type of life can be found living in there. And again, Margarita said, well, let's try to capture a shrimp. Actually, maybe we can get the chance of getting such shrimp in the patchy rock we're looking for, which will be in the next stop after this push core.
Yeah, it's really rocky underneath. Are you happy with that top though?
Let me see the profile, please. As long as it's 5 centimeters, it should be just fine. I'm not gonna.
You happy with that?
This is just fine, yes. Thank you so much.
And now to the rocky patch. And I have a good comment here. Maybe Nick can comment on the comment on Porter D. Pressures only affect animals with gases in them, like fish with swim bladders.
There's a question. Pressure only affects animals with gases in them, like fish. That is correct. That comment is correct. The pressure will affect animals only with swim bladders. If you try to if you try to catch a fish and take it to the surface, it will be, the swim bladder will expand because of the gases, and it's going to have a very adverse effect on the fish.
Unless you took the stuff.
Things like invertebrates, echinoderms, copepods, crinoids, they don't get affected.
And then someone commented, I would love to have a shrimp cocktail. Actually, it was the case today. Today at lunch, we have a shrimp cocktail. So we were joking, hey, don't eat my samples. Of course, those are the shrimp you can buy in the supermarket, right? So it was kind of funny to see those guys that were seen there in the screen now, shrimps everywhere, and then having shrimps also for lunch.
Yeah, sorry, I answered on the forward one.
Right.
I got you.
The comment on the pressure that only affects animals with gases is correct, but if you dig a little deeper, actually it's I believe the answer is a little more complex, especially for people that maybe looking at gene expression, but it may not be appropriate for this light discussion that we have here. But again, depending on what you're looking for, okay? So, but again, the comment is correct as is.
So Margarita asked me, what is your personal opinion on shrimps? Well, actually, I like them, but prefer living in an environment like this one, not in a plate. Oh, actually, Austin, you're making also a very good comment. We have actually we have 2 chefs on board in this expedition, and actually we have pretty good food, which is a worry for many of us that are trying to get left weight on us. Actually, the food is quite delicious. So again, someone asked, well, any of these, but the ROV pilots or people from the Schmidt Ocean Institute that were on their home, which is the RV Falcon 2, have they reef tanks at home?
What's a reef tank?
Fish tanks. Fish tanks. Or with
Armando, is this the rock garden you were talking about?
not yet. Those are the fish. The rock garden is rocks, as you know them. Eran roca puras.
Okay.
Tipo basalto.
Oh, these are the ones.
Okay, so we could go to the right side. Those are the pectoral rock we're looking for. So we could get down as low as you can. So start zooming on those rocks. Yeah, it's very similar to this one. So since we are there, we will have more time for sample. For example, look, let's start zooming in this one first as a beginning, and then looking for everything else. Right. Now we got this one.
Right, we're gonna be stopping here.
Yeah, so we are seeing now, maybe Rick
Okay,
can comment where all the living things we're seeing around. There's several.
I agree. Yeah, so right now we're looking at the white, the small white, let's say worm is actually a sea cucumber. It's kind of a protected on this basalt, right? Okay, and then you see, what kind of coral is that? Blue? So it's an octocoral, and then a crinoid on the left side with many ophiurines sparkled everywhere, and then on the very left hand side you will see an acorn worm, hemicordata. I don't know what is this. Oh no, that's just these are other rock formations.
Yes.
Okay. For the yeah, let's see. We're looking. The for the comment on the sounds of reefs, this is very correct. This is a very hot area of research. Apparently, a very healthy reef has a very different complex sound profile versus a degraded reef. So there's a lot of work that is going on right now.
Nick, and what we are seeing here?
This is we're looking at the podia of a sea cucumber, and we're looking at
And the ones that are on top of the rock that you have just in front?
Can we zoom in a little more?
That's all full.
Okay.
But I can bump up and get closer to the
Okay, that was good.
Yeah.
Which one? You told me. Let's look at that limpet.
Puede ser, could this be a brachipod?
We We don't know.
Would you like to take any of this rock entirely? For example, this one or
We
this
can-
one?
It could be a limpet.
See?
We should get it if it is
Yeah,
possible.
can we get the entire rock if possible? Or this one maybe?
Position, get closer to it.
Yep.
But we should
Thank you.
zoom in. Guillermo.
Yeah, no, I know, but it's not like it's there. Yep. Where do you want it?
In what space do you have available for taking rocks with animals?
Hay de temperaturas. That corner, that first one.
If it's gonna go with a rack, in any biobags.
BB1C?
B1C, okay. Yes. BB1C, Antonio?
We already have something in there, but I guess it
We could go there as
is
well?
different enough so
Okay,
we don't mix them up.
okay.
Yeah, that's what it said, BB1C. Yeah.
So what is this, Nick?
We do not know yet
Okay,
exactly
so a rock
what it
with
is.
some kind of
It's-
unknown
it's-
organism.
It's possible that is. Oh. Oh. No, that's a point. All this time I've been. Okay, so let me be corrected. The white specimen is a polychaete, not an echinoderm. Okay, and this is what science is about. If you're wrong, you have to admit it and move on.
Almost. Oh, it's escaping.
You see here live the challenges
Yep.
of try to collect at those depths. These mechanical arms are unbelievable, yet there's a lot of difficulty, especially when it comes to smaller organisms. Yeah, I think it looks like that. Is that small? It's not a small, is it? Just check this one out. All that effort. This one, so. Guillermo, can I tell you it's smaller? There you go.
It's, as you can see, it resists to be sampled, that rock. It's a pretty stubborn rock. Okay, so last try.
Last time, yeah.
Let's try. We know we're going to get the coral that is just in front of us.
Okay.
There.
Can you answer that? If it's
Yes,
I don't know.
he did it.
What? Oh, the sediment. Is the sediment organic in lean nature or primarily inorganic with some organic content? So we believe that most of the sediments are inorganic, but they have lots of organics on top of them. So it's a mixture that some of the organism in there filters, taking the organic matter on them and leave them almost sterile, right? But yeah, most of them are a mixture. ¿Puede ser un coral reef? Puede ser el 4, el 6, el 7 o el 8.
So now we're going to collect the white coral that you found for in coral quiver number 4. So now we're going to collect the coral that we just saw in the background there. Just below there you can see it. We're going to collect it in a special container called the coral quiver. And yes, success. And then the chart says resistance futile rock.
So this
That's
one here, I think 4 is,
forest, basically
6 is open.
4,
coral 6,
6, 7, or 8. All of them.
8's got a, did
Six,
they
six.
put a plastic bag in it or something this morning?
Yeah, I think the bag is there, yes.
So 6 is just the, number 6, so it's just 6, 47 seasons.
Oh, don't worry. Thank you to share the program. That's what we're here for. To share our science with all of you.
6 or 7.
So we also have some greetings to give here to Fabian Poblete, one of the students of Professor Guillermo, and also Titzli Castillo. Oh, we are allowed to 7:30. Yes, of course. That's next. Okay. So now they're asking in the chat, is that rover arm made of titanium or is it an alloy? It is made of titanium, yes.
There's a question if the robot arm is made of titanium or is it an alloy?
Titanium.
It's made of titanium, yes.
Do you want the whole thing or just
Titanium.
From the base.
It should be easy to dislodge. It should be very easy to dislodge.
Are we okay to put that in the bio box?
Yes, that would be fine.
A pie box or a coral quiver?
It's probably
Whatever
bio
is
box.
easier.
Whatever it
Okay.
is.
Okay,
We're running
bio
out of
box
time, so
is
better-
easier.
yeah.
Okay. Which one? Maybe in the BBT2B?
BB2B?
BB2B, yep.
Okay.
You see the polyps have, that all the tentacles have been retrieved. you could actually count the tentacles and you will find 8 tentacles, which is a taxonomic character for that defines octocorals, let's say cousins of the hard corals. All Cnidaria. Cnidaria include the jellyfish, the corals, the soft corals, okay, just the sea anemones. Daniel, saludos, profesor Guillermo. No sé, Daniela Prison.
Well.
Alright, which one? Yeah.
We are going into file box 2B, which is right there. Is that one up here?
Along down.
So we have a greeting also from Daniela for saludos, Profesor Guillermo. Guillermo says thank you very much. Yeah, Billy back too says, this is crazy. Actually, it is. It's always fascinating, as my friend Spock would say, to inspect new environments in where you actually don't know what you're going to find. And also some greeting for the engineer. Yes, they know how to do their stuff, huh?
We have 15 minutes left. I suggest to look exclusively for brachiopods.
Yeah, that's a
maybe.
good idea. Actually, I'm seeing just pointing here to a rock that seems to have them. So we may zoom on it to see that is the case. Yeah, could we zoom on that rock in order to zoom in that rock here, where they may be populated with brachiopods? No. Sponge. Maybe we move a little bit, 1 more meter down. We look zoom in, 1 meter down, zoom in, and so on. Left?
On the whitish rock.
Here?
Yes. Oh no, that's a gastropod. That's a gastropod.
You want it?
No, brachiopods. We would like that.
But not now.
Verdaderamente no tenemos tiempo.
Okay. So again, hop 1 meter, zoom in rocks.
Is it a mollusk?
Gastropod, yes, a mollusk, yes.
Maybe here.
It's actually very difficult to know right now.
Nick, what about here? Nothing, right?
This is a crinoid. This is a small crinoid, but I don't see anything that.
What about the thing I'm pointing?
I don't know. Nothing that is.
Nothing like look back up here, huh?
Nothing that is very obvious. Oh, sorry.
Okay, so I would suggest to hop in a few meters and then get down again and inspect a new set of rocks.
What do they look like?
They look
Like small bivalves.
like this. 1 centimeter.
Oh,
very
right,
small.
okay. Like a, yeah, they're flat, right?
Yeah. Maybe going in that direction.
Yeah, sure.
So again, we're looking for these ancient organisms which are brachiopods. Can you share the link for the ROV page, please? Yes, of course. Maybe Sophia can do that. Thank you.
We're gonna zoom in and we'll just go right.
Yeah, let's zoom in and then Nick, whenever you want, say.
Right in the middle of the screen, there is. That's a sponge.
Okay, sponge. Let's keep going.
Let me get there a second.
Yeah, come
Yeah.
here.
10
Come
minutes.
here.
What about upper left there? Upper left of the screen. The white thing, is that it?
I'm looking. Let me do it, yeah.
Oh,
You take what? This one? This
Yeah.
is a sponge.
That's fine, okay.
Yeah.
Yeah, I'm just gonna fly nice and slow, so you just hang out
Yeah.
here, just let us know if you see anything.
So now with me is Nick directly in front of the skin with a pointer saying, hey, here we may have a
So that's
brachipod.
the white polypid again.
Right.
I don't see where? I don't see anything obvious.
No, that not?
No.
Okay, let's keep moving.
Hexactinellid sponges, beautiful. I haven't seen them before. Look at this tube, eh?
Mm-hmm.
No.
It's a dual.
Tubo, sí. Yep. Yeah. We don't. Right now, there's no time for that.
The brown one up there, is that?
But here?
To the left there.
Again, look and see
No,
all the
these are
animals
the
you are
one
looking
for-
around also.
Okay.
From far it seems there is nothing, but then again,
This is a stock animal.
snail.
This, can we just zoom in on this? Which is probably a sponge, but I just want to.
Yeah, it's sponge.
It's a sponge, okay. What is this? It's a little.
Can we zoom in that?
No, you're fine.
Pardon, okay. Is it another snail? Yeah.
It looks like a snail, right?
That's a snail. Really interesting.
What about those?
No, they are extremely small. Extremely small.
Okay.
But you can take the rope.
You were getting this, which was,
Okay.
is the size.
Okay.
We had like a.
Oh, thank you for the comment. I always enjoy these streams so much. Thank you for guys for making this possible. Again, for me at least, it's no. Yeah, let's get that.
Let's get this. Let's get this. It's maybe a bivalve, but it's the right.
For me at least, there is no sense of making science if you cannot share with everyone, right? It does not make, for me, it not make sense
Armando?
to learn new things if we cannot share it with everyone interested.
We may have a 2.5 liters napkin bottle at some point for Miguel. Whenever.
And also Drew.
And also another one for Drew.
I think Drew wants 5 liters.
Okay.
Let's see. I don't know. The one that we're looking was definitely a bivalve light. That's the closest we have been
Okay.
so far.
Yeah, it's right in front of us. Now.
Okay. Yeah.
Is that one, right?
Yeah, it's right here.
Wow.
It's like a clam.
Yeah, but they can be like that. I mean, this is the closest we've been.
Okay,
so far.
let's get it.
It's probably. I doubt it's.
Let's have a look at that other one up here too.
Okay.
So, this one, where was it? Right there.
Broken shell.
It's into
Yes,
a broken
broken
shell.
shell.
Yeah.
So they're very white in color then.
No.
Not necessarily.
Grayish,
Yes.
gray,
Okay, gonna select that one?
Yes, please.
So we are on 2. You want to be, I guess we'll go to a fresh one here, so we'll go to 3.
Yep.
Oh, we have never been to.
Drew 2.5 and Miguel 2.5.
No. Drew.
Carlos.
Drew wants a 5 liters?
No, 2.5.
We
Okay.
have,
Katie just confirmed.
Okay.
we have
2
an
meters, right?
hexactinellid expert
At
in the
the
audience.
surface.
He's calling the two sponges of the genus Caulophycus.
Okay, so let's keep looking for similar things that look like may look.
Anything that same
Brachiopods?
size.
Yeah, let's. I'll tell you what. I'll take off and we'll just go nice and slow. And
Okay.
Keep the camera zoomed in.
Okay.
Just if you see something like.
Can you do one for sample and includes place where the start?
Possibly bivalve or brachiopod. I don't think it's a brachiopod, but it will be.
Can you confirm me the suction sample number?
Number 2. That was the number 3.
Okay. Thank you.
Always on rocks.
Always in rocks.
Yes.
Let me zoom out here and just.
2 meters over the surface is fine.
No, no, no. What about
Yeah,
over that
2
cross
meters.
field?
Yeah, when the water is
Yeah.
clean.
No, no. Uh,
Look, then let's go back there where there are rocks.
Two risky barrels.
2.5 for Miguel, 2.5 for Katie, 2 meters high when the sediment is settled down.
Okay.
Pero lo último
Yeah, the ultra.
Okay.
Yeah, this one. But I don't know. That's right. That's a little better. They need to be cemented or they have a pedicle on a rock. Right. Yeah. They cannot be on a.
I'm gonna just slide over here. That one looks like it's on a rock for sure.
Surrounded by probably it could be a sponge or it could be paranos. Yeah,
I'm
sponge.
just gonna set down the zoom.
Ooh, could be. Yeah, I don't know. It's just like a laying flat.
It's definitely flat on it.
Yes.
Do you want us to grab it?
Yes.
Yeah.
Okay, and I'll just have a quick look at this one.
And then that one.
My eyes just.
Oh.
Also that one.
No, that looks very, very strong. Oh, no. I see. I don't know if it's my eyes.
Uh-huh.
My eyes.
It looks like it's.
Yeah.
Okay.
I start making bivalves out of sponges with my eyes.
Yeah,
so
I know.
that's not good.
Wishful looking.
Yeah, wishful looking. Yeah.
Okay, so I'm just gonna bump up and we'll grab this one for I'll move over to the forearm. How do you want to grab that?
That's the last arm. And then there is the others.
Maybe
I'll stay here.
They'll probably put it over in. You want to back to this one too?
Yes.
Yeah. So it's right here.
We're going to BB2B.
No. And it's going to go into that same box. Sorry, 2B.
Yeah. Actually, our next mission we're planning with Dr. Drew Palmer. It will be a mission to the moon. But then you have to wait for that a little bit.
We're going to grab the shell, not the echo.
Interplanetary Armando, yes. Thank you for that. And then we are having the sample one and also a free worm also in the right. So 2 samples per one for the price of one.
Perfect. That's That's very good. That's as good as you can get.
It can get not more efficient than that, huh?
You have 3 for 1.
3 for 1, right?
To the line up to
Maybe in the same bio box, the next one.
Yep.
So in the same box, the next one.
that's okay. The next one is about the bubble over too.
They look like the only thing is, yeah, they look like a sponge if we.
But I don't think it's sponge,
I
right?
think it's a sponge.
It is a sponge?
Yeah, I think it's
Okay,
a sponge.
not
yes.
this one.
I agree. Yeah.
Let's just kind of cruise over the rubble field here.
Okay.
And we'll, now that I know what we're looking for.
So they are those Calophacus. These are the Calophacus hexactinellid sponges. Thank you. An audience member seems to be very well versed in hexactinellid taxonomy.
I think you saw one in the
They're getting to us way too small. Way too small. This is like a,
Yeah, Liz is making a comment to live in the European oceans. Actually, we don't know what we might find there. We suspect at least we have all the ingredients required for life in the oceans of Europa. So there may be a good chance of finding microorganisms, but who knows what else can we find there. Since science is based on evidence, we will not know until we go and explore, right? So again, this mission is just like going to the oceans of Europa, because we are exploring the unknown. We are a group of experts here fixed to a screen looking for the unknown.
not right there, that. Yeah.
Sonic
That
will
has
just
the right
detect.
shape. That seems to have the right kind of texture. Yeah, let's see that. Yeah. Oh, I got my eyes. Yeah.
I don't think.
No, that's a sea anemone. The closer
It's-
I get, no.
It's a sea anemone, okay. So in advance, when we're about to finish, we're going to collect one more push core for one of our research scientists whose friend is interested in nematode, this very small animal that looks like worms. We're trying to look at the
This
sediments.
to the left, probably a sponge, but
That's a sponge.
it's
Sponge.
always they have the same shape, they have the same color.
That's a sponge.
And then before leaving, we're going to take some water samples for the people that are interested in that as well here. What is in the water column, so to speak, in the waters just above the surface here. So those were going to be our last samples of today's diving.
Look at that.
Yeah, that's an anemone.
Yeah.
It's the same thing as before, yeah. We're getting triggered by anything around this.
What is that?
That looks like it's definitely not it has a pentameter symmetry, so some kind of a juvenile echinoderm.
Right.
That's a gastropod.
Okay.
Yeah, that's a gastropod.
You want it?
What? That's another sponge there.
Punch also. So we're discussing here where there are so many little animals that looks like the one we're looking for, but they're not. So
also you're getting an idea of the hidden biodiversity.
Yeah.
If you just
in just that's a very good comment. In just a few square meters, look at all the lives we're finding.
These are probably no, these are tentacles. They're probably ascidians.
Mm-hmm.
Maybe a hidden sea urchin, but it's
Okay.
It's not that white one above the sponge.
Oh, they no, they're not ascidians. They are sea tentacles, sea cucumbers, but that's a sponge.
On the right, have you seen the animal on the right?
The two, eh?
No, on the right. More, no, up. Ah,
That-
that one.
That looks like a sea anemone to me. Absolutely, that's a sea anemone. Yes.
It's a sponge there.
Yeah, that's a sponge. That's probably a tanade.
So you may see now the guys that we're looking for, the brachiopods are not easy to find.
So that's a bivalve.
And the
Could be, it could be interesting. Is this? I'm wondering if this is alive or
No,
it's
just a shell.
if it's just a cell. It could be. They have ridges sometimes. Some.
Let me take off. I'll just keep cruising up here.
Okay.
Some brachiopods have ridges like this. Right now I cannot tell what it is.
Yeah, you're making a very good comment, James. At the meter plus scale, when you look at it from that, it looks like a garden of rocks only. But when you get close to it, it's full of little animals, right? You can see, yeah, one of the surf you already collected at the left. And also squat lobster just below the white bright thing.
I'm looking at I see what you're looking at.
Maybe we want to spec this group of rocks in detail.
Yeah. You see the crinoid to the left. Oh, several crinoids on the upper.
A garden of crinos, huh? And again, there's many species of crinos that we know in the fossil record. And we are seeing the living relatives of those here.
That's the crab there.
Oh, now we're zooming in on a crab. What type of crab is that, Nick? Squat lobster.
Oh, yes.
This one?
This, yes.
Okay, this one. It seems we have found one of the brachiopods. It looks like one, huh?
Looks like one. Yeah, it looks like one because I say.
So we're trying to collect the one that just have at the left that looks like a round, creamy color thing that we're zooming on in now. And it seems it's a brachiopods, right?
Yeah, that looks very good actually.
Okay,
That looks very good.
Okay, let's collect that one then, and then we're going to take the Niskin bottles, then the core, and then we leave. James asked a very interesting question. It is because the volume to surface area relation. Maybe Nick?
Are we grabbing this?
Yes, this one. Maybe Nick or Guillermo can comment on that? On the relation between the volume to surface area relation, these little animals we are seeing here? I just said that. I just said that. Right? I just said the skin buttons and the crossroad theory. There. Perfect.
Eso es. Eso es.
We're going to put it back at 3, this guy here.
Okay. Give it to me as well. Okay, here now we wait a little bit to sediment to move around and then we take the Niskin buttons, please. How many?
2 of the 2.5.
Yes,
Okay.
2.5.
2.5.
Yeah.
Okay.
2B. Niskins?
Yes, 2 Niskin bottles, please.
What do we have here? We have, I think, all 6 of them.
2.5.
2.5?
Yes. And then one last push core with the top 5 centimeter and that's it.
5 and 6. 2.5.
Just the 2.5 or?
2.5.
Okay. 2.5. 5, 6.
¿No hay pizza? No, no sé, ¿eh?
Sí, después tenemos reservado. Y él también.
Bananas and kiwi.
So number 5 and 6.
And push core you said? Which one up?
Yes, one push core for Lauren.
I'm gonna have to bump out into a better spot because there won't be
Yeah.
anything here.
That will be our last activity of today's day.
Okay. I think you got 13 or 15, whatever. 13, 14, 15, 16. Or 17.
So we have a comment here from the moment that these organisms are primarily concentrated near the belt seeps, but in this case we have not detected such bent or seeps. So how they survive, maybe Nick can comment
Yes.
on this? How these organisms survive since we don't see any evidence source of our energy like seeps or vents?
The question again was, how do they survive? What about, I would assume that the organs are primarily concentrated near the vent shapes. What about regions like, no, I have already answered that.
Okay.
I have already answered that, yeah. That's the answer there, yes. We answered that too.
Okay, perfect.
That's all answered.
Thank you. So James is asking what happened to the yesterday jaws. You changed it, right? Because yesterday we were using yellow ones and since we were using red ones. Well, they tend to break as it happened yesterday, so you have to replace it from time to time.
Yes, this is close. Yesterday jaws were broken, so they had to be replaced.
So Nick commented that yesterday jaws were broken, so they had to be replaced. Yeah, that's a good place for Lawrence
One last chance to make it good.
core.
I have a good feeling that. I have a good feeling.
The pilots, are the jaws 3D printed on board?
The inserts are 3D printed on board, yes.
The pilots mentioned that the jaws are 3D printed on board.
And there goes our core for learning, being the last activity of today's dive, which has been pretty productive.
One more.
Yeah.
Okay.
PLA? No, it's PLA.
Outside years?
That's for Lauren, correct?
Yes.
Okay.
So that's Lawrence core for looking for nematodes.
What is this PLA?
Sammy is asking whether we don't have trouble with vibration doing 3D printing on board.
That's what?
Type of
No.
printer.
Plastics.
No problems at all.
No troubles with Just give us a minute and the pilots will answer the questions. They're very busy right now. Try to put the core in the right place.
Well, you already answered. I already answered that question. All right, so with this, we finish our today's dive, as I said, with a lot of samples and analysis we're going to take in the next months and all the live rocks and everything we took.
Last question for the pilots. They are asking, I think, for what kind of material people are using. We are using to build those jaws. This is nomenclature that I do not know. ABS or PETG. Or
For the jaws, TPU.
TPU.
It's like a rubber compound.
TPU. That's the material.
Yes.
TPU.
And with that, I say thank you to everyone.
And the vibrations do not interfere with 3D printing.
And with that, I say thank you to everyone and for your company on today. As I said, we love to share our science with everyone. And of course, as a big thanks to the pilots and operators. They are the one that make this possible. Again, thank you and goodbye, everyone.
Yeah. You want to know what it is right now?
Hey everybody, good evening from Falcor 2. Thank you so much for tuning in for ROV SuBastian's 679th dive. As always, we'll be launching again tomorrow morning, and yes, Ascent stream will be on. And we all invite you to join us tomorrow. Just as a quick review, today we transected the shallower portion of Site 9. We were hoping to see some methane seeps, but we definitely saw some interesting geological formations and sampled some myofauna, as well as various microbiological organisms. So, pretty successful day if you ask me. And with that, I'll sign off for now. We're going to have our night team of marine technicians come in to watch the Ascent stream for you. So huge thank you to them for all their support as well. And with that, I wish you all a really good night. Take good care and see you tomorrow.
Every time I'm like, oh, you're gonna love this stuff.